Our group performed the soil, water, and ash-free dry mass, or weight experiment. This experiment analyzes the water content and organic content found in the soil. Soil, water content, and ash-free dry weight are forms of metadata that help in analyzing microbial community diversity in soil. Soil water is essential to microbial activity, and different textured soils hold on to variable amounts of water based on clay and organic content. The higher the clay and mineral content, the higher the water content. Gravimetric determination was utilized to compute water content in the soil as well as the ash-free dry weight. Water content is a proxy for organic matter content of the soil, which is the ash-free dry weight, calculated by subtracting the ash weight from the dry weight of the sample. Because of the reliance on gravimetric determination, high accuracy is necessary when weighing the soil samples, which are weighed using an analytical balance with accuracy to 10 micrograms. The methods used to determine the organic matter content of the soil in this lab was by drying the soil, then combusting and oxidizing organic matter to carbon dioxide at an elevated temperature in a muffle furnace, with the weights being recorded after each treatment. The ash-free dry weight indicates the total organic content in soil and can help indicate taxonomic abundance and diversity.
The soil samples used in this experiment were collected from two different locations. Samples P1, P2, and P3 came from just off of a dirt road in Kuntz next to some large pine trees, while samples B1, B2, and B3 came from a yard in Beaumont. After being collected, each soil sample was sieved to separate the coarse particles and extraneous materials, and the sieved soil was used as the starting material for this experiment. The soil from the Beaumont location was black in color with an appearance similar to coffee grounds, and the soil from the Kuntz location was a lighter brown color, as seen in image A. 18 total labeled foil pans, 3 for each site, were used to hold the soil, and these pans also served as weighing boats. A scoopula and an analytical balance, similar to the one seen in image B, were used to obtain high accuracy mass measurements of the soil. A single-wall transite oven and muffle furnace, seen in images C and D respectively, were also required for completion of the procedure.
Measurements of soil, water, and organic content can be collected through a two-step procedure consisting of a drying period followed by incineration in a muffle furnace to obtain the dry weight and ash-free dry weight of the sample. To begin the procedure, the foil containers used to house the soil samples were labeled and then weighed. Next, 3 roughly 10-gram samples of previously sieved soil were added to the foil containers and the weights recorded to the 6th decimal place. The samples were then loaded into a drying oven at 105 degrees Celsius for a period of approximately 24 hours. After drying, samples were briefly removed from the drying oven, weighed, and then returned to the oven to prevent moisture accumulation. The dried samples were then transferred to a muffle furnace set to 360 degrees Celsius for a period of 2 hours to oxidize organic matter within the soil. After cooling, the weight of each sample was recorded to the 6th decimal place. Final values for water weight and ash weight were calculated by subtracting the dry and ash-free weights from the wet and dried soil weights, respectively.
Here you can see how we placed all 18 of the foil pans into the single-wall transient oven, careful to leave space between each row of pans so that one would not mistakenly knock any over.
After remaining in the oven for about 24 hours and in the muffle furnace for 2 hours, the soil samples are shown to have lost their original color due to the intense heat.
So Table 1 holds the original measurements made before any of the calculations. So the foil and wet soil samples were collected and measured before being placed in the drying oven. The foil and dry soil samples were collected and measured after being in the drying oven, but before being placed in the muffle furnace. And the foil and ash samples were collected and measured after being placed in the muffle furnace. Table 2 shows the weights in grams of the soil after subtracting the weight of the foil pan from the initial combined measurements from Table 1. The wet weight, again, which was taken before drying, the dry weight taken after drying, and the ash weight taken after combustion are all reflected in Table 2. The water content in grams was calculated by subtracting the dry weight from the wet weight. This calculation represents the amount of water that was present in the soil prior to drying. The ash-free dry weight in grams was calculated by subtracting the ash weight from the dry weight. This calculation represents the amount of organic matter that was combusted in the muffle furnace.
Table 3 represents the percentage of water in the soil and the percentage of ash-free dry mass in the soil after removal of the water. Table 4 shows the means of each of these percentages for each sample. From initial glances, it looks like there was a possible larger percentage of water found in the B sample, and the ash-free dry mass percentages look similar. It is difficult to tell if these samples are significantly different without further calculations. Table 5 shows the standard deviations for the water content and ash-free dry mass percentages of each soil sample. Table 6 represents the 95% confidence intervals for the water content and ash-free dry mass percentages of each sample. As seen in Table 6, none of the values are below the alpha value 0.05, so none of the values are significantly different. This means the water content and the organic content found in the B and P samples are not significantly different.